because our families were close, and Doug would take his kids down to Lytle Ranch in southern Utah, right at the corner of like the state where it gets in the Mojave Desert. And he took me along with them. I tagged along on some of those trips, and they were just like hugely influential. I knew like you could do this. You can like chase lizards and snakes around and find them, and that's you call that a job. Uh, so when I went to college at Utah State University, I found the local herpetologist, a guy named Butch Brody, who studied lizards and snakes and frogs and salamanders and stuff, and I just kind of like inserted myself into his lab and I never really looked back. I kind of knew from my experiences with Rob's family that this was like a thing you could do. I don't know why Rob went on to study plants rather than like animals, but yeah. And I was Rob's older than I am. I was actually really close with Rob's younger brother John, who is also a professor. He's a professor of psychology now. Rob's two other brothers, Ken and Trevor, are just like an engineer and a lawyer, so they're like the losers. You can tell them I said hi. But yeah, so I worked with Brody in his lab on flat-tailed horned lizards in Arizona, and that was cool. But there were sidewinder rattlesnakes there, and I spent all my spare time like obsessing about sidewinders. You can track them in the sand. They have a unique like sand trackways you can follow. I tracked lots and lots of sidewinders and would watch them sitting in their little craters, doing their ambush foraging thing. And so when I went to grad school, my advisors, Harry Green and Craig Adler at Cornell University, they just gave me like free reign. They were like, you could study whatever you want, come up with some interesting questions. And so I kind of thought back to my experiences doing this, like, work observation stuff with sidewinders. And so I kind of glommed onto that as like, studying ambush foraging behavior. And I'm not very imaginative because I've never really switched away from it. Like, my idea, I just kind of kept pursuing like similar things. But I did learn in that process. And one of the things that I love best about academia is that you do have intellectual freedom. Especially in a university setting, you can kind of study whatever you want. And it's not like the university cares what exactly you're studying, as long as you're being some sort of a productive scientist. And so I've really taken that and just kind of spent my time in my career with my own grad students going after different kind of questions that I find interesting at the time for whatever reason. So this is kind of a sampling of things that are going on in the lab now and in the recent past. This is from a poster I put together for the department a little while ago. So we do various things, a lot of which center around still kind of the feeding ecology of rattlesnakes. And so that's what I thought I'd talk about today. Is I'm just going to draw from some projects that have been going on that all kind of center on this ambush foraging behavior of rattlesnake species. And if you're not familiar with this, I'll give like a brief overview. Viperine snakes in general and rattlesnakes in particular are extreme ambush predators, so they're sit and wait predators. They spend the vast majority of their time in a coil hiding somewhere where they think prey can come close enough that they can strike out and eat it. So this makes them extremely boring from a behavioral perspective, because this is like a time-lapse video of a snake. If you look right up there in the corner, you can see the prairie rattlesnake coiled. And it's doing what it does. This is like 99% of their surface activity is this. And you can kind of see like the desert is coming alive here in the morning, but the snake never moves. It doesn't like flinch a muscle. It's like extremely patient and cryptic, and this is kind of their strategy for trying to get something to eat. And so they'll spend days to weeks kind of sitting in one kind of area, and then they might give up and move to a new area, but they're waiting for something to come close enough that they can try to bite it. So here's that moment. This is a sidewinder with a desert kangaroo rat that's like sidled by at night. This is like extremely slowed down to show that what the snake tries to do when prey comes close enough is just launch its head fast enough and accurately enough and open its mouth at the same time to project the two fangs that are usually folded up against the roof of the mouth forward to try to embed the fangs so it can inject venom, bite that thing, and then let it go. They almost always have a strike and release strategy, especially against like bigger prey, like small mammals. They'll bite, inject venom, let it go, and then the animal wanders off and dies somewhere. So this is a snake the next morning that has tracked the carcass of a kangaroo rat that it bit, and it's finally like reached the carcass of the animal, which can be like many meters away from where it was bit, and you know it's going through the process that snakes are also famous for, which is being able to ingest relatively huge things. So it can, you know, all the bones in the skull are really kinetic and movable, and it can like walk this thing down its gullet and get like a huge meal that it can will then last it for a long period of time. It can go off and digest that thing. At some point, when it's done digesting, it'll start the cycle over again when it's ready, and there'll be like a period of kind of active search for prey cues where the animal's moving around on the surface again, trying to find somewhere to set up an ambush coil. And it's using chemical cues and probably mainly vocal cues and olfactory cues, but maybe also some visual cues. And if we're thinking about the visual system of snakes, we also have to think about temperature, because they famously have pit organs where they're able to see infrared radiation. So the pit organs of snakes are these like membranes that are suspended in the cavity of pit vipers that are integrated with the optic tectum in the brain. So they're specialized brain regions that take input from the pit organs, and then they feed into the optic tectum. So it's long thought that a snake, a pit viper, sees a composite image of these two things. This is a squirrel filmed with a thermal imaging camera, which uses exactly the same principle as the pit organ, transducing thermal contrast in the environment into imagery, and that's probably integrated with like whatever they're seeing from the eyes into like one visual field. So I became interested in this because this is from our ground squirrel work with snakes. We did for many years. We worked on predator-prey interactions between California ground squirrels and snakes. They have this really interesting kind of signaling interaction, And I'm showing this with like, you know, this integrated with this with thermal imaging camera, and I'm doing something that I recognize we probably shouldn't do, because thermal imaging cameras are like lens-based camera systems that focus radiation onto a very sensitive receptor. Pit organs are not like that. They're like a very shallow kind of pinhole camera aperture that does not have the ability, presumably, to focus radiation. So people do this all the time and use like thermal imaging in our technology as a shorthand for what snakes might see, whereas biophysical models of snake infrared perception indicate that like a mouse on a log a couple meters away might look like one of these images, which isn't really like a spatially resolvable visual image at all. So there's kind of a disconnect between what we can see with thermal imaging cameras and what snakes may or may not be able to see in some sense. And I wanted to explore that because with all this work we were doing with the ground squirrels, at the same time we were doing kind of work looking at ground squirrel rattlesnake interactions and how like the squirrel signaling behavior affected the snake. One of my co-authors on some of these, Don Owings, one of his students published this paper on the potential of ground squirrels using the tail as like an infrared signal when they're waving it at snakes, because he found that they tend to have like warmer tail temperatures when they're signaling towards rattlesnakes compared to gopher snakes, which are not pit organ bearing snakes. So presumably can't see this. So they interpret this as potential evidence of like an infrared signal. Part of this signal to the snake was in the IR spectrum. And I remember being in a meeting with Aaron and one of George Balkins, who's collaborating with me on this project, one of his students, getting into a big argument about this because Georgia's student kept being like, the snakes don't see that. They can't see like that. They're not resolving that type of detail. I don't care what the squirrel looks like, and the snakes aren't going to see it that way. And we'd actually done some work. I had a student who did some work with kangaroo rats showing kind of the same thing. That if you look at the body temperature of a kangaroo rat in great detail with the thermal imaging camera, its body temperature does change after it interacts with the rattlesnake, a potential predator. It shunts, temperature around to various places. The legs get warmer, the head gets warmer. But we kind of interpret that as, well, that's probably just kind of autonomic nervous responses to this heightened state of vigilance. The snakes may or may not be able to see this. And when I started asking people like George, he was like, well, yeah, we just don't know. Like, nobody's tried to crack into like the inner workings of the ability of the pit organ to resolve spatial details. So George had an idea to like come to San Diego State, and since I had a colony of rattlesnakes, it's like, I'll help you build this device in your lab, and we'll figure it out. We'll build like this custom snake vision testing device where we can present to snakes. So the idea is we put a snake in a box in a completely darkened room, so there's no ambient light at all. We have a background plate that we can control the temperature of, and a little stimulus target that we can also control the temperature of, that is released from an electromagnet. So when we cut power to electromagnet, the thing just silently swings in front of the background. So with the thermal imaging camera, it looks something like this. When the camera is sitting where the snake is, there's just like a warm target moving in front of like a homogeneous thermal background. What we're looking at is the snake, which is sitting in the box facing it. And this is one of our study subjects who is going to watch a target swing in front of it. And so when I play this video, you'll see the pendulum. And I don't know how well you can see that. Maybe I can turn this off. The snake basically showed no response. So the great thing about rattlesnakes is that most of the time they sit there very absolutely still. Whereas if something interesting goes on, they almost always flick their tongue at it. So here's the same snake like 2 minutes later after we increase the temperature of the target and release it. And you can see there's just like some subtle head movements and tongue flicking behavior there. Indicating to us, at least, that kind of evoked behavior like, oh, well, that snake probably detected something. And so we do that enough times with enough kind of stimulus targets, enough different temperatures with enough animals, and we start to build out kind of the parameters of what the snakes can and can't see and what affects their ability to resolve a specific target. So we kind of build the proof of concepts. And then just recently, so this was published like several years ago. Ever since then, we've been working on this basic question of what happens when we make it harder to resolve the spatial details. And the way we're doing that is we're presenting a target in front of a gridded background that is not homogeneous. It's a heterogeneous gridded background. And the target is the temperature of the average of the warm and cool spots in the background. It's a contrast sensitivity threshold experiment. People in like visual research use this all the time to compare visual acuity, like spatial visual acuity across different mammal species and bird species. It's basically the same as can you tell that this is black and white versus does it just look gray to you? If you're sitting in the back of the room and you're looking at this one, you probably can't tell that's actually black and white bars. You can't resolve the background, so you see it as the average. Another way to look at it is with a thermal imaging camera that's defocused. So this is our thermal imaging camera not in focus, and there's a pendulum swinging in front of a gridded background that you can't really see until you focus the camera to resolve the background. Once you can resolve the background, then you see clearly that, oh, there's actually like a target moving in front of it that is just happens to be the average temperature of the background. So what we did is we took a bunch of backgrounds of different spatial frequency, larger and coarser and coarser grid sizes, basically, from 8 to 38 millimeters, and presented like isothermal targets in front of them to see like when can the snake actually resolve the background and start seeing the target. That will tell us something basic about the spatial acuity of its IR system. So just to give you a sense, that should change with the contrast. So the contrast between the cool parts of the grid and the target, it should be easier to resolve if that's higher contrast. So we did this at a number of temperature contrasts. So from our previous study where there's no grid, you get something like this, where you get like much stronger evoked tongue flick responses on the part of the snakes at these higher contrasts. And so here I'm about to show 3 years worth of data across these grid sizes, just so you can appreciate how long it took my team of undergrads to do this. Basically, the snakes do not respond even at the coarsest grid sizes. Statistically, if we look at, do all our modeling, there's a bunch of like non-significant results. It's like they can't resolve the target even at the very coarsest grid sizes, which we thought should have been within their ability to see. So they really do not have like good spatial resolution systems. I was worried that they just kind of habituated over the course of this prolonged experiment. So we actually ran like a post test, and they haven't habituated. If we take the grid away from the background and retest them again, they can very easily resolve the details. So that kind of tells us where we are right now is that we've kind of confirmed that George's original biophysical modeling is probably correct. There isn't, we thought that maybe there was like a central nervous system processing mechanism. There's a couple papers that indicate that neural networks could sharpen imagery with a certain type of like central nervous system processing, and people thought maybe snakes were doing that. I don't think they do. I think their visual system, their spatial resolution is much more like this versus not like that. There are some caveats to this. Like everybody else who does like pit organ work with snakes, we're doing this in complete darkness, so the eyes are essentially non-functional. I do want to change that to see if there's potential synergism between the eyes and the pits, but I don't think that's very likely. So I'm not actually sure how to think of the pit organ system. Maybe it's not so much an imaging system. I'm talking with a neurophysiologist in Germany, Tobias Kohl, about looking more about like kind of the threshold at which sensory systems like activate other sensory systems. Maybe the pit organ system is more of like a motion detector, like an alarm system. Like it has a much lower threshold for response, so that snakes that are extreme ambush predators that spend all their time just sitting there, like pit vipers and like boids and pythons, which also have independently evolved these pit organ thermal detection visual systems. Maybe it's somehow involved in kind of responding at a lower threshold to potential stimulus and bringing other sensory systems online. I don't know. There's other ideas that we're trying to figure out, so we don't really know where to go with it. But I do think we're missing something kind of fundamental about the pit organ system and its kind of functionality. So maybe I'll have more updates on that in the near future. But regardless, and I'm not arguing that it's not important for the predatory behavior. I still think that it probably serves some fundamental function for nocturnal predators that are preying on endothermic small mammals at night. It does, what is certainly important, and we've seen this in field studies, is that snakes are very careful about selecting ambush sites that appear to have homogeneous thermal backgrounds. And now I understand more why that might be so important to them, is it heightens that sensory system. Okay, so if they can actually manage to strike something, I want to say talk a little bit about venom and venom injection, because another thing that came out of the ground squirrel study, this is something that always bothered me with the ground squirrel study, is the snakes, like venom is like this linchpin of their system. Like in order for them to kill and eat a live animal, they have to inject venom in it, and the venom has to work because they almost always let it go. That animal has to die, and then they find the carcass and swallow it. So if the venom isn't effective, then the animal's not an effective predator. They can't constrict. They're not fast. They can't like actively pursue things like coach whips. And ground squirrels are famously very physiologically resistant to snake venom, and so are wood rats, another local species that's eaten a lot by one of our local red diamond rattlesnakes. And it kind of bothered me that there's this disconnect between what their prey can do and what we keep pulling out of the stomachs of the snakes. Red diamond rattlesnakes and western diamondbacks, to some extent, like rely on wood rats as like their main source of energy. And especially in more northern areas of California, western rattlesnakes or Pacific rattlesnakes rely on ground squirrels. You know, that's what we find in their guts all the time. And so, like, why are squirrels or wood rats only like partially resistant or sometimes resistant to snake venom? Why aren't they more resistant or resistant enough to be able to survive bites and not end up in the stomachs of snakes? And so I finally had a master's student, Kelly, who decided to tackle this and to just like look across like wood rats and squirrels and southern Pacific rattlesnakes and red diamond rattlesnakes and kind of compare inter and intra-specific variation in venom resistance across all of these. So it was very ambitious, and I'm super impressed that she pulled it off in the course of her master's work. Because she had 2 snake species and 2 prey species, which both have like a strong species interaction, but then will occasionally also prey on the other mammal. So, you know, we'll find wood rats in western rattlesnakes and squirrels and red diamond rattlesnakes sometimes. So there's some like cross-species predation going on there. And so she actually looked at this at population level. So we had different populations of small mammals and snakes that she was pulling from. And then at the individual level, within those populations, we also looked at individual variation associated with different mammals. And what she found was like a lot of variability. So if you look at red diamond and southern Pacific venom versus like ground squirrels, some populations of ground squirrels are really not resistant at all to like western rattlesnake venom, and some are much more resistant. And not, this is all in San Diego County, so these populations are not very widespread. This happens at a very small spatial scale. Wood rats, these southern Pacific Mission Trail rattlesnakes have metalloproteases in their venom that are basically not inhibited by any of the small mammals. So I'm calling this venom resistance, but it's actually the mechanism for the small mammals resisting snake venom is to inhibit the metalloprotease fraction of the venom. So there's a fraction that's responsible for kind of digesting tissue and spreading out other components of the venom. And that's what the small mammals are resistant to. So that's what we assay, is kind of metalloprotease, the ability of the serum to inhibit venom metalloproteases when the venom isn't pooled, but it comes from specific snakes at specific locations. And I should back up and point out that like they do have fairly different venom. So there's a pretty good metalloprotease fraction in both species, but it's much more diverse in red diamond rattlesnakes. They have a whole bunch of different flavors of metalloprotease, if you will. Whereas southern Pacifics have like all sorts of kind of odd, smaller peptides and other molecules that are active in their venom. They're not so reliant on the metalloprotease fraction. I should also point out that being resistant to the metalloproteases of southern Pacifics did not in any sense mean you would also be resistant to the metalloprotease of red diamond rattlesnakes. So these isoforms of different metalloprotease enzymes vary enough that it's very biologically significant. This is just like individual animals, you know, being resistant to one is basically not predictive at all of being resistant to the other metalloprotease snake fraction. So not only do components of the venom vary, but even within kind of a class of toxins, there's a lot of variation. And we did recover a signature that wood rats are just repeatedly less resistant to the local red diamond rattlesnakes than they are to allopatric red diamond rattlesnake populations. So that's a kind of indication to us, at least on some level, that red diamond rattlesnakes are probably evolving venoms to continue to be effective in the face of the evolution of resistance of their preferred prey or their primary prey. They seem to be able to maintain effective metallopermease fractions, at least against their local populations. We also found this one is interesting to me, that difference in metallopermease fraction for the Western rattlesnakes or the Pacific rattlesnakes, it really only happens where they overlap with red diamond rattlesnakes. All the populations we have on hand, at least. As soon as you move in the range of Pacific rattlesnakes, as soon as you get out of the area of overlap with red diamonds, they have much more typical metalloprotease dominant venom. They go back to like what other rattlesnake species usually have. So there's potential. This is one thing that I want to follow up on. There's the potential for like character displacement within predatory snakes that are large-bodied and eating small mammals. I think there's even some potential that maybe the coevolutionary dynamic between red diamond rattlesnakes and wood rats means that it might be more optimal for this other snake species to shift to kind of reliance on a different type of venom than the typical metalloprotease venom. And then on top of all that variation, there's a huge amount of individual variation within the population of squirrels and wood rats. A given individual at a given time might be highly resistant to the metalloproteases and snake venoms, or might not be. And that, at least for one of our populations of squirrels, we had a bunch of data on body condition having to do with the size of the squirrel and kind of stress hormones that were present in the squirrel. And we could see a pattern between low body condition and inability to inhibit the metalloprotease fraction in snakes. So I do think that maybe there is we don't actually know what the costs are to them expressing this metalloprotease inhibition in the blood serum of the small mammals. I'd love to know more about it. We haven't actually even identified those molecules. We know that they're small peptides, but we don't know a lot else about them. So we have, Kelly's thesis has opened up all these other questions about like the plasticity of the mechanism, what it is, how plastic it might be, and what trade-offs come with expressing it. I think it could be a really, really fruitful kind of coevolutionary system to understand kind of costs and benefits with these types, expressing these types of anti-predator adaptations. So I'm trying to, we had like some pilot funding for Kelly's thesis, and now I'm trying to actually get like more major funding to continue to look into that. So we'll see where that part goes. But the other thing that came out of the whole like venom injection, our small mammals resistant to venom thing, is initially when we were doing like the behavior work with ground squirrels and kangaroo rats, we also had a project, with ground squirrels and rattlesnakes, we had a project with kangaroo rats and rattlesnakes. And as we were watching them, I kind of thought like, are kangaroo rats resistant to snake venom? Because they seem to be able to survive these snake bites. Like we were watching our K-rats and we'd see things on our field cameras like this. About to step in front of that sidewinder, where both the animals are moving so fast that with like a traditional camera, we couldn't see what was going on. It's like there's a blur of motion, the snake crashes into the rat hops off and is fine. Whereas usually when we see that type of thing with our field snake watching cams, the small mammal dies and the snake goes off and finds the carcass and eats it. And so this happened enough times that I convinced our venom collaborator to run some kangaroo rat serum. It's like, these things have to be resistant to snake venom too, right? And he's like, no, there's nothing, like they don't seem to be resistant at all. And so then I was like, well, are the snakes just like missing somehow? So I convinced one of my other collaborators, Tim Hyam at UC Riverside, to loan me some high-speed cameras from his lab. And we had some pilot data to think that this kangaroo rats were doing were essentially dodgy, but we needed to look at it more carefully. So we rigged together this setup. This is my students, Grace and Malachi, who did most of this work, where we'd have to haul out like these huge battery packs, many IR lights, 2 of these high-speed cameras, rig it all up in the desert at night where snakes were foraging so we could get kangaroo rats to come in and film one of these interactions in situ. It was ridiculous. It was like 500 pounds of equipment that we were like hauling around from like spot to spot to set up and film this. But it did work when it worked. Like, you know, we'd go many nights between getting an interaction. But when we found it, we'd see something like this because we're watching at 500 frames per second and we could see it from like the two different camera angles. So we could reconstruct like what happens in 3 dimensions. And it was indeed like when snakes contacted the kangaroo rats at all, it was usually the kangaroo rats were moving so quickly, so rapidly that they would just power right out of the way of the strike. So if you've seen these, it's because Grace and Malachi published a few years ago now, and they also put together like a public outreach site, Ninja Rat, to like show all of our snake predation videos. So I'll just kind of quickly summarize the basics. This is really interesting to me to kind of look back and see, okay, here's what happens most of the time with kangaroo rats and snakes, or at least for this species. This is Dipodomys deserti, the desert kangaroo rat. So it's like a sand dune specialist that overlaps with sidewinder rattlesnakes. Maybe about 20% of the time, the snake is targeting and either misses or is somehow like overshoots. The kangaroo rat doesn't dodge, the snake just misses. That's what this looks like. This snake would not have hit that kangaroo rat even if that kangaroo rat hadn't moved. So we would call that like an inaccurate strike. A little over 25% of the time, the kangaroo rat just moves so quickly, its reaction time is so fast. Incidentally, it's probably hearing with their enlarged auditory bullae the sound of the snake uncoiling, and they can jump so explosively that it just takes them out of the path of the snake. All this is like happening in under like half a second. This is the snake strike goes from like still to fully extended in like 0.6 seconds on average. And you know, those explosive jumps are so powerful, you can see it just like takes the kangaroo rat like rocketing through the night sky. And sometimes the power of the jump just kind of pushes the snake away. You can see this one just, the snake head was kind of in contact, but it jumped. The explosive jump kind of pushes the kangaroo rat so quickly through the path of the snake can't embed the fangs. And even if the snake does embed the fangs, so this is about half the time we're calling a bite when the snake actually manages to get its fangs into the kangaroo rat. And about half the time when that happens, the kangaroo rat is actually able to dislodge the snake like mid-bite. So here's a sidewinder that actually gets its fangs embedded. And the kangaroo rat does like a twist jump kick maneuver and sends the snake like flying away. It gets its powerful limbs like there and like kicks the snake off. So that leaves the poor snake with only, oh, and this one, I've got to show this one because it just kind of shows kind of the weirdness that can occur sometimes. So this is a kangaroo rat that's going to jump out of the way of the striking snake. And if you watch carefully, it kind of slips. It doesn't get its foot under it. So the snake actually manages to reach it and get its fangs embedded. But the kangaroo rat does one of those kicks while it's falling. And you can see it gets a foot like in the mouth of the snake. And it's kind of keeping the mouth of the snake out with its hind feet. And once the snake is kind of dislodged, then the K-rats have enough time to like reorient themselves, and this one goes like jumping away. So that means only about 25% of the time the snake actually manages to control the kangaroo rat enough that it gets fangs embedded long enough that it can actually inject enough venom to kill the prey. So this is an example of that. It looks like this goes on for like a really long time, but keep in mind it's under a second. You know, the snake is latched on, and you can kind of see it. It doesn't get kicked off. In this case, the snake is doing the very typical pit viper release. So when the snake is injected venom, it just pulls the fangs out and kind of recoils like that. And really the only time we ever see kangaroo rats that don't like just run away and land on their feet and go bounding away is when they've been injected with like every dose of venom. That's when it flops on its side and doesn't get very far before it dies, and that's how the snake gets its meal. Anyway, so that answered our question as to like how are kangaroo rats like surviving these bites, and also opened up lots of other interesting questions that we had about kangaroo rats and rattlesnakes. And that's where some of my research is going now, is delving kind of further into the relationship between kangaroo rats and rattlesnakes, because, because I like it, and I want to do it, but also because kangaroo rats are very important in kind of the ecosystems that they occur in, and they are, they have a really strong species interaction with rattlesnakes in some of these populations. This is data from Andy's thesis, Kristen, who in the sandhills in Nebraska, like Ord's kangaroo rat is like by far the dominant portion of the diet of prairie rattlesnakes there. So, they're not, I don't know how many times Ords have to strike unsuccessfully, or prairie strike at Ords to kill them, but they do it enough that it's a very important part of their diet. And so we're actually focusing on a prairie rattlesnake Ord system now. These are two of my current students, Brian and Jessica, who are working on this project. And again, the idea is, so there are lots of landscapes in the West that are obviously like impacted in a dominant way by kangaroo rats. Kangaroo rats are kind of a keystone species. This is like the Carrizo Plains in California. All those like mounds you can see, those are the mounds of the giant kangaroo rat. You can see it's just like extensive, like across this whole area. They build these huge burrow systems. They are granivorous seed cachers, so they eat lots of plant seeds and move them all over. There's all this like old ecological work from James Brown's lab showing that, if you exclude kangaroo rats from one of these arid ecosystems, there's like immediate profound impacts on the vegetation and other aspects of the community. And so we kind of thought, well, we can use their interaction with rattlesnakes as a way in to kind of argue that we should understand more details about how these animals potentially impact each other, because rattlesnakes are so numerically dominant as predators in a lot of these ecosystems. This is our site in the Marathon Grasslands. These are like 50 adult rattlesnakes that we found in that one summer that we marked and released, and we were constantly finding new ones. I mean, if you look at good population estimates for a lot of rattlesnake predators, it's like hundreds of adults per square kilometer. Like there's in an area like this, you might find like one or two kit foxes or something like that. Other small mammal predators are there, but they're not nearly as common as some of these like low energy ambush foraging rattlesnakes. So our idea here is we're actually looking across like the range. These two species are kind of co-distributed over a broad range in the Midwest. So we have sites in Texas and Nebraska, and way up there in Alberta, the northern edge of the range of Ords and prairie rattlesnakes. And we're trying to kind of test this idea that's out there in the ecological literature about potential asymmetry in how ectotherms and endotherms might respond to changes in temperature, rapid changes in temperature associated with climate warming. Especially like nighttime temperatures in the northern part of North America are rising really rapidly. And since both of these animals are nocturnal, we think that could potentially have a profound effect on the way kangaroo rats and rattlesnakes interact with each other. So I'm working with these two other labs. We're focusing on the behavioral part of it to try to understand the basic impact of temperature on things like the spatial ecology and the seasonal and diurnal activity patterns of the snakes and the kangaroo rats. Tim's group is working more on biomechanics and muscle physiology, and then we're working with a computational modeling group that is potentially going to be able to take a lot of our field data and try to make sense of it and maybe simulate what might happen as the species interaction changes due to changing temperatures. So that's kind of the big picture, long-term view. In the short term, what we're doing is I've hit on this idea of using animal-borne accelerometers in this system as kind of this key tool to give us this really fine-scale, detailed view of what kangaroos and rattlesnakes do on a day-to-day, night-to-night, hour-to-hour basis. And you can tell a lot. This is becoming really popular in like the mammalian carnivore literature and other areas of the wildlife literature, using these animal-borne accelerometers to try to say something about like the behavioral profiles of free-range animals. For those of you who aren't familiar with it, the idea is that you have a triaxial accelerometer that's attached to the animal, and when the animal is doing kind of a species-typical behavior, it logs a typical pattern of XYZ acceleration. And then you use complicated machine learning models because it's logging this, at like 20 times a second for all three axes, or 25 times a second is a typical like logging pattern. So you're going to pull like millions and millions of data points off these accelerometers, and to make sense of them, you then use a machine learning model framework and a bunch of validated behavioral observations to tell your computer model, hey, this is what the animal is doing at this time. What does that acceleration pattern look like? And eventually the program will learn the typical acceleration patterns associated with typical behaviors is the hope. And then you can use that data to tell what your animals are doing when you're not even watching them and they're running around wearing their little accelerometry backpacks. We experimented with a bunch of different ways to try to get this on a limbless animal, and it came down to tape. Surgical tape was the best thing. This like tegaderm tape adheres the best, and it adheres long enough and well enough that we can tape like a little accelerometry backpack onto a prairie rattlesnake, and it will carry it around. And then we recover the animal is radio tagged, so we can recover the accelerometer later, and it will download all the data to tell us hopefully what that snake was doing. With the kangaroo rats, it's a lot easier. We just have like a little backpack. They're only about 2 grams, the accelerometers, and so it's right there while this guy's digging up or caching some seed. You can see it wiggling around this little accelerometer. And the ones on the snakes look like this. So there you can see it's kind of taped on the back of the snake. We use slightly bigger ones that will last longer. The kangaroo rat one, the battery on that thing will last maybe 4 days, and then we have to recover it. And the snake will last for several weeks before we have to recover it. So we can get like a longer watching frame. We tried to validate a lot of the snake foraging behavior with these field cams that we use for a lot of our field stuff, where you mount a camera to watch the snake while it's sitting in ambush. This is one of our prairie rattlesnakes wearing their little backpack. So we can get data that way. But then we also have to, you need a lot of validation data. So we've been doing some captive behavioral studies over the past year to look at this as well. So I can show an example of that. And it's really kind of feeding rate. We really want like gory detail of like feeding rate from the snakes. And so we want to be able to validate striking and swallowing. Those are the behaviors that we are key to whether or not the snake, not only did it bite and kill something. So here it is striking one of our lab mice. And striking is easy. That's like a gimme. So here's the data from the accelerometer for the 10 seconds, like 5 seconds before and 5 seconds after the snake struck the mouse. You can see this obvious huge burst of acceleration associated with the rapid body motion of the snake. And then, that snake sits there for a while. They actually usually have a quiescent phase after injecting venom. And then you can see it, starts lateral undulation as it moves around, potentially looking for the carcass of that mammal. So let me play this whole sequence. This is going to be sped up like 20 times. But there's the strike. There's the snake sitting still. Now the snake starts moving, that you can see the accelerometer is picking up the motion of the snake as it moves around. And eventually, this is a simulation of what they do in the wild. They follow the scent trail of the animal, kind of spend some time investigating the carcass. They often will kind of drag the carcass around to get it in a position where they can swallow it. So we key that out. And then here it's starting the ingestion process. So it's kind of using its head, and the accelerometer isn't up near the head, so it's probably not going to be able to pick a lot of this up. But it's kind of walking the carcass down. We are getting a really good signature for when they swallow the carcass. If you look carefully, you can see this bolus that is going to go down the gullet of the snake, and then as it passes under the accelerometer, there is luckily kind of this unique wave signature that happens that we think will be key to us picking this up with our field snakes. So most of what we've done has actually been in the lab. We've only piloted like a few like free-range and prairie rattlesnakes that have these accelerometers. We did get one of our snakes striking something though. So we have like one validated like field strike so far, and we're hoping to get a lot more. We've been, looking at the data in various ways, and it's obvious if you look at like the overall dynamic body acceleration as a summary statistic that's often used in these models to differentiate different behaviors. And we do have different behaviors, and we've just started using some of the machine learning approaches. And as you might predict, like striking is like 100% recall precision and accuracy. It's like obvious to the machine learning model to pick up like a strike. What is great to us is that swallowing is actually really high as well, to differentiate from either the snake just sitting there or the snake slithering. You have to build these models iteratively and have a lot of validation. So we've only started with some of the basic behaviors, but it does look really promising. With the kangaroo rats, we've done a little bit of this as well. Just moving or not moving is obviously comes through pretty well. But even what kind of general foraging behavior has pretty good precision and accuracy. The recall isn't as good. But I think that can improve as we get kind of more validated kangaroo rat behavioral data sets and build like a more complicated version of the model. And we'll also be able to maybe differentiate a bunch of other key behaviors like explosive jumping or burrowing versus seed caching. We'll have to also kind of simulate some of that with kangaroo rats in enclosures, but a lot of that, the kangaroo rats are actually great for just watching them. If you're careful and you have like a night vision camera, you can kind of sneak up on them and just watch them for several hours of the night. So we hope to get a lot of our validated data sets that way. And then again, the hope is to bring this all back into this complicated modeling framework eventually to understand kind of the basics of the interaction and how that interaction might scale up into population or ecosystem level parameters. So we'll see where that goes. So that work, we'll actually be working out at Monahan Sandhills this summer for our Orange Kangaroo Rat team. This is the Orange Prairie team that's potentially going to be out there this summer. If anyone who's really interested in seeing our field setup in action, I guess it's only about an hour and a half from here. So drop me a line and you come up and check us out. And I threw this up there just to say thanks. Like all of this work was done by like large teams of students. Who are, typically I have lots of students working together to throw a lot of person hours at the problems for this type of field work. So, huge thanks to everyone for all their efforts in assembling the field portion of the data. I think maybe we still have some time where I can talk with you guys, answer any questions. Yeah.
Great talk. Thank you for coming out. I was impressed that you managed to do as much as you did with the metallic proteases because they're a wily and frankly vexing group to work with.
Yeah.
I had a thought, don't know if it's worth anything.
Yeah.
Look at the TIMP family of peptides.
Okay.
They are much more generalized. You'll have 4 TIMP regulate something like 26, 28 matrix metalloproteases. They're easy to genotype, and their varies. The genotype does vary with gene expression, so that's easy if you assay that. That's easy to get.
Yeah. Yeah, we haven't actually used any of those like gene expression tools. I'd love to. This was the assays for this were all done in like fluorescent plates, like plate readers and digesting like a fluorescent substrate. So pretty basic.
Last reason, the TIMs are heavily associated with rapid tissue regeneration and the inhibition of matrix metalloproteases. So it could be a connection there with how you're seeing that resistance.
Okay, that would be really interesting. I'd love to crack into that more. Thank you.
How polymorphic do you find some of the venoms, especially in prairies? Notoriously polymorphic, and we actually caught some down around Post years and years ago, milked them and sent the venom off to someplace in Utah, and they analyzed it and came back and said, yeah, you've got neurotoxins, you've got this and everything else in there. So
I think that's more the norm than we realize for a lot of these snakes. Like they have all sorts of variation within species, especially wide-ranging species geographically, and from what we can tell just locally in San Diego, that can vary over a pretty short spatial scale from population to population. I think they're very rapidly evolving and maybe differentially expressed toxin families. So it's really complicated. And I don't want to become like a biochemist, so I'm always going to do this collaboratively and try to like focus on like the snake part of it. But I do I just think it's like a really interesting coevolutionary dynamic. Yep.
So with the metalloprotease stuff, so when it's inhibited, it's blocked, or are there a range of injuries that these rats and squirrels are suffering? I mean,
so when it seems like for the animals that are highly resistant, when it's inhibited, they don't show symptoms from some of the other characteristics of the venom. But that's based on like watching whole animal responses after being bit by snakes. So it's not, we haven't actually done like more detailed, studies of like exactly how much damage is done. If we'd have to like sacrifice the animal and do some sort of autopsy to see like, was there some localized damage and the squirrels just kind of shake it off. I did repeatedly see squirrels surviving what looked like very good bites from snakes, and I knew that they were resistant, and so I always assumed that the snakes are probably getting a fair amount of venom in here. That's just a resistant squirrel, but then other squirrels would succumb and the snakes would eat them. So, that's the whole thing that started the project off, was kind of this repeated variation. And so I assumed that maybe there's other molecular mechanisms. The metalloprotease inhibition is just the one that people have found. And I've talked to a couple of the venom guys who have looked for other things and haven't found like another mechanism of resistance other than metalloprotease inhibition. So I'm not sure though. I think the venom is so complicated. There could easily be lots of other things going on.
So on her study, was it in Orange County?
She had, all of her samples are from San Diego County, but then we borrowed some samples from Bill Hayes' lab, who was in Orange and Riverside County.
So all the 4 sites were the same type of ecosystem?
Yeah, they're all like coastal sage scrub, California grasslandy type ecosystems where we could find, we pulled all 4 species from each of our 4 sites. So we identified sites that would have enough of all 4 of those species where we could look at them all sympatrically and then compare them allopatrically. Yep.
The accelerometer signals based off of the environment
Well, the accelerometer signal, all it's doing is just picking up movement. So it's just logging acceleration like 25 times a second. So whatever the animal it does, like responding to environmental cues or whatever, accelerometer is like a really basic like piece of electronics in some sense. It's just logging the movement, and that's it. So it doesn't. People do build like other. Our accelerometers actually do have a thermistor in them. It also logs temperature every 5 minutes, just kind of independently, and that's important to us because of our interest in ambient temperature. But they're just logging like movement or not movement. So if the little piezoelectric crystal is being moved forward, it's like accelerating or decelerating, or it's just like sitting still. Yeah.
I'm just fascinated with your talk, but some of the basic ecological relationships are really helpful to me, such as which states really exert a population control on those, and then how do the snakes find the right ambush spot?
Yeah.
And then how does the snake avoid being put out in itself?
Well, the ambush spot, again, I'll answer that one because I know more about that one than the other two. That's mainly chemosensory cues from what we could tell. So it's just tongue flicking around, and I think they're probably investigating the environment and maybe integrating like, other information, visual cues, etc. No, they tend to they'll find a site and they'll stay there for several days. And if it's a desert snake, they'll seek thermal refuge during the day, come out at night, and post up somewhere in that same area again. This is like typical. Obviously, there's variation here, but then they'll move. Yeah, so they'll be in front of burrows. They'll be next to mammal trackways. They'll be sometimes in an area where I'm like, why are you here? I can't see anything obvious that looks to me like it's a prey trail, but then lo and behold, like a little mouse runs by later that night. So I think that's mainly chemosensory cues. In terms of control, I feel like we need to know more about kind of the direct and indirect effects of snakes as predators on small mammal populations. I think there's an equilibrium. I think like in a lot of predator-prey systems, there is some sort of equilibrium. And that's what we think potentially could be disrupted. I mean, maybe not. Maybe they don't have that much impact on small mammal populations. But we'll chase them around and take data on it and hopefully be able to say something about it eventually. I forget what the third thing.
Well, the problem is just the basic ecology of what we're going to find. This is going to predation.
Yeah. So I'll tell you this. I have a student who's working on like a note right now because with our snake cams at night, we have like thousands and thousands of hours of like rattlesnakes sitting, like waiting in ambush on the surface in various species at various times. We have never once seen one killed by a predator. We've seen predators come into the camera somewhat frequently, and they almost always kind of investigate and leave. So I think that, rattlesnakes do have predators. They are predated by things. But I think it's almost always they're most vulnerable when they're stretched out and like moving between sites. It seems like when they're in this kind of ambush coil and they're like either cryptic or coiled and a potential predator comes to investigate, it never escalates. They don't, I think the things that do eat them are picking them off during the times when they're not like hunting, from what I can tell. Either that or they're just like really, really infrequently predated.
So those slides with the pie diagram we were going through.
Yeah.
So the very first one.
Yeah.
What was up with that kangaroo rat? It's got black on it.
Oh, that we had to dye mark them. That's us. To tell the individual kangaroo rats, we use this dianesyl pelage dye.
Oh, okay.
So because we were doing like behavioral studies on kangaroo rats, yeah. So that was totally unnatural. Yeah.
So, Toby, a great point about when they're avoiding predation. If you went, you have to go a little bit more east, but if you went where Crotalis and eastern indigos overlapped,
Yeah.
You could look at that because when you're hunting, you're typically using your best sense. But when you're afraid or when you're detecting a possible predation,
Yeah.
you're using all your your senses. So it might give you some, slightly different.
Yeah. Yeah, they're definitely like, it's kind of front of our mind that if you're a kangaroo rat, there are times that if you have an ectothermic predator, there are times of year and under certain environmental conditions where you probably don't have to worry about it, where they're just not a threat because they're not active and you can be. And so that's one of the things that we're hoping to kind of take more quantitative data on in terms of how some of these like indirect effects of predators may or may not affect kangaroo rats. Like if they start encountering lots of rattlesnakes and it's summer and they have to spend more time like being careful of watching where they're going, spending more time vigilant.
Right.
We're hoping that the acceleration patterns of like basic movement that we can actually experimentally like scare kangaroo rats and see if there's like basic activity patterns that change as a result of that for some certain amount of time as an indication of living in a landscape of fear type of thing.
Right.
So
those Eastern, I was unclear about this, I apologize. Those Eastern indigos, they will actively hunt rattlesnakes.
oh right, yes, that's right. They're the rattlesnake predators.
Yeah.
Yeah, I was thinking kangaroo. They were eating
Yeah, I'm
kangaroo
sorry.
rats. Yeah. Oh yeah. That's true. I've seen like videos of them like chomping the heads of like copperheads. And, yeah. So I didn't realize they overlap with prairies in some areas. Huh?
Yeah, you'd have to, edit a bit.
Yeah.
But there's certainly other crotalus species.
Yeah. So that would be one. We've never worked in an area that has like an abundance of like an ophidiophagic snake. So California king snakes don't actually eat rattlesnakes very often at all. It's like a really minor part of their diet, but the indigo snake might be another story. Yep.
Yeah, these different species that you work with, what is the chemotoxic versus neurotoxic composition amongst them? Is it almost entirely chemotoxic, or how much?
The Southern Pacific rattlesnake in San Diego County and in other areas, again, where they seem to overlap with red diamond rattlesnakes, they have neurotoxic components in the venom. Some of those like peptides are probably neurotoxic. We don't know for sure though what they do. Like most of them are kind of, people are guessing that they have like various neurotoxic effects, but like the particulars of the identification of those toxins aren't worked out the same way they are for like Mojave rattlesnakes, where we have this famous like toxin that varies from population to population.
So with the current rattlesnake, is it mainly cytotoxic venom that you're seeing the destruction of cells?
Mainly, yeah. They're mainly like the typical like metalloprotease dominant, lots of other kind of tissue destructive hemorrhagic type of proteins and enzymes and stuff. But it does vary. Like geographically, I think it depends on where you're getting your prairie rattlesnake from. So we're collaborating with the Venom lab on that for the prairie stuff. There's a guy, Steve Mackesy, who is taking like all our venom samples to look in more detail about like variation throughout the range where we're collecting. That's not specifically tied in with the K-rats, but it's just, when we knock a snake out to like process it, we always take venom because why not? It's, we want those samples for like downstream use.
No questions from the chat? Any other questions? Well, thank you. And we'll also have this uploaded on YouTube and have the presentation as well. So keep an eye out for the email if you're not.
Sure. Yeah, nice to meet you. Southern Africa, most of the time, I talk about